Hello, friend. Welcome to Science for Sleep. Whether you have joined me many times before, or you are discovering this quiet corner for the first time, this is a place to slow down, learn something gentle, and allow the day to gradually fade into the background. Tonight, we are going to look closely at one of the most familiar fruits in the world. A pineapple seems simple enough when it is resting on a kitchen counter. It has a heavy golden body, a crown of stiff green leaves, and a rough surface covered in repeating diamond shapes. At first glance, it appears to be one large fruit, much like an apple, an orange, or a peach. But a pineapple is not one fruit in the ordinary sense. It is a structure created from roughly 100 to 200 separate flowers. Each of those flowers once occupied its own small position around a thick central stem. Each one had petals, pollen-producing parts, and an ovary capable of developing into a small fruit of its own. As the flowers faded, those small fruits began to grow beside one another. Their tissues expanded until their borders became pressed together and joined into the single solid pineapple we recognize today. Botanists call this kind of structure a multiple fruit. That simply means one large fruiting body has formed from many flowers belonging to the same flower cluster. If you enjoy these quiet journeys, you're welcome to subscribe. And if it feels natural, to leave a comment sharing where you're listening from tonight. It helps others find this calm corner of science, one sleepy listener at a time. The cultivated pineapple has the scientific name Ananas commosus. It belongs to a tropical American family of plants called the bromeliads. Yet even among its relatives, the pineapple has developed an especially unusual way of building its fruit. A peach begins with one flower. Inside that flower is one ovary, the structure that later enlarges around the seed. An apple also begins with one flower, even though several parts of that flower contribute to the finished apple. A pineapple begins differently. Instead of one flower producing one large fruit, an entire crowd of flowers works together. The clearest evidence is still visible on the outside. Every raised diamond on the rind is commonly called an eye. Each eye marks the former position of one individual flower and the small fruitlet that developed beneath it. The eyes are not separate scales placed over a smooth fruit. They are the remaining surface pattern of the many fruits that grew together. Between the eyes are shallow grooves, where neighboring floral units once met. Those units became so closely joined that cutting into a ripe pineapple reveals almost continuous yellow flesh. The original borders are still present in its structure, but they are no longer easy to separate. The flower ovaries are only part of what we eat. The finished pineapple also contains tissue from the bracts, which were small leaf-like structures beneath each flower. The sepals that protected the flower bud also contributed material. The thick central flowering stem enlarged as well, becoming the firmer core that runs from the bottom of the pineapple toward its leafy crown. This is why a pineapple is more than a bundle of tiny fruits pressed together. Many kinds of tissue took part in the merger. The fruitlets expanded, the bracts became fleshy, the central stem thickened. The spaces between the flowers gradually filled until the whole cluster behaved like one connected body. The botanical word syncarp is sometimes used for a fruit formed through this kind of fusion. It may sound technical, but the idea is easy to picture. more than 100 flowers once opened separately. They then grew so closely together that their individual fruits became parts of one larger structure. The rough rind preserves the map of that transformation. The sweet flesh hides most of the boundaries, and the crown remains attached to the same central axis on which all those flowers first appeared. To understand how a plant can assemble something so complicated, it helps to move below the fruit itself. There, beneath the golden body and sharp green crown, is a low tropical plant with no trunk, no branches, and a remarkably compact way of gathering the energy needed to build its many-flowered fruit. The pineapple rests on a thick stalk rising from the center of a low circle of leaves. No trunk or woody branch supports its weight. The plant grows close to the ground as a large perennial herb. A perennial is a plant that can remain alive for more than 2 years, rather than completing its entire life cycle during a single growing season. From above, its leaves form a rosette, with each blade arranged around the same central point. This pattern resembles the overlapping petals of a wide green flower. A mature cultivated pineapple plant may spread between 1 and 2 meters across. With its fruit and crown included, it can approach a similar height. The exact size depends on the variety, climate, soil, and age of the plant. Its rosette commonly carries between 30 and 50 mature leaves. Some of those leaves can grow close to 1 meter long. Many narrow to a sharp point, and some varieties carry small spines along their edges. The leaves angle outward from the center, allowing them to occupy a broad area without needing branches. Together, they create the main surface where the plant collects sunlight. Most of the structure holding those leaves together remains hidden. Beneath their tightly overlapping bases is a short, thick stem. The stem does not stretch upward like the trunk of a tree. Its growth is compressed, so one leaf attaches close above another. This tight spacing helps produce the dense rosette visible at ground level. New leaves begin at a small growing point in the center. Young leaves appear there first, folded closely together and protected by the older leaves around them. As they lengthen, they gradually bend outward and join the wider ring. The same central growing point will eventually change its task. After the plant reaches the right stage of development, it will stop making ordinary leaves and begin constructing the flower cluster that becomes the fruit. The future pineapple therefore begins deep inside the rosette, long before any flower or fruit can be seen. The plant belongs to a family called Bromelaceae, usually known as the bromeliad family. Most bromeliads are native to tropical and subtropical parts of the Americas. The family contains more than 3,000 known species, ranging from small plants that fit inside a hand to large ground-growing forms with leaves several meters long. Many ornamental bromeliads are grown for their bright red, orange, pink, or purple flower structures. Others are air plants, including species in the genus Tillandsia. Air plants often grow on tree branches, where they use the tree for support without drawing food from its tissues. Plants that live this way are called epiphytes. Cultivated pineapple follows a different pattern. Its roots normally grow in soil, and its heavy fruit develops close to the ground. Even so, it shares several useful traits with its bromeliad relatives. The leaves have a tough internal framework made from strong bundles of fibers. These fibers help the long blades remain upright even when rain, wind, or their own weight pulls against them. A waxy outer coating covers much of the leaf surface. This coating slows the escape of water and shields the living cells beneath it from intense sunlight. Some pineapple leaves also curve slightly upward along their edges, creating a shallow channel that can guide rain or dew toward the center and base of the plant. Beneath the soil, the roots usually remain fairly shallow. They absorb water and dissolved minerals from the upper layers of earth, where heavy tropical rain may arrive quickly and then drain away. Good drainage matters because roots still need access to oxygen. Soil that remains saturated for too long can damage them and create conditions that favor rot. Above those roots, the entire rosette works as one broad solar surface. Sunlight entering the leaves supplies energy for photosynthesis, the process that uses carbon dioxide and water to make sugars. Those sugars support every new leaf and root. Much later, a large share of that stored energy will be directed into the central stalk and its crowded group of flowers. The crown, rind, sweet flesh, and firm core all depend on work first carried out across these long green blades. Their shape, fibers, wax, and close arrangement make the plant a compact system for gathering energy while holding onto water. Inside the leaf surface, microscopic pores help control that balance through a daily rhythm that becomes most active after darkness arrives. Along the lower surface of each pineapple leaf are thousands of microscopic openings. These openings are called stomata. Each one is bordered by two curved guard cells that swell or relax, changing the size of the pore between them. Carbon dioxide from the air enters through these pores. That carbon becomes part of the sugars the plant builds during photosynthesis. Water vapor can escape through the same opening. A leaf therefore faces a constant trade between collecting carbon and holding onto moisture. For many crops, most stomata open during daylight. Sunlight is available then, allowing carbon dioxide to enter, while the plant uses light energy to build sugar. This schedule works well when water is easy to replace. A pineapple often grows under conditions where conservation matters. Its wild ancestors developed in tropical South America, where exposed leaves receive intense sunlight and rainfall can be strongly seasonal. Warm, moving air speeds evaporation. A long pineapple leaf exposed to heat and wind could lose considerable moisture while gathering the carbon needed for growth. The plant reduces that loss by shifting much of its gas exchange into the night. Pineapple uses a system called crassulacean acid metabolism. The name comes from a plant family in which the process was studied early, and scientists usually shorten it to CAM. After sunset, pineapple stomata open more widely. Night air is commonly cooler, with higher relative humidity. Under those conditions, less water escapes for each amount of carbon dioxide that enters. An enzyme inside the leaf helps attach the incoming carbon dioxide to another molecule. This reaction produces organic acids, especially malic acid. Malic acid also contributes to the tart flavor of apples. Although inside a pineapple leaf, its main role is temporary carbon storage. The acid is held inside large compartments called vacuoles. A vacuole is a fluid-filled space within a plant cell. By dawn, millions of leaf cells contain acid made from carbon gathered during the night. Daylight changes the process. The stomata close more tightly as temperatures rise. Water remains inside the leaf more effectively, while the stored acids release carbon dioxide within the cells. This internal supply enters the Calvin cycle. the group of chemical reactions plants use to build carbohydrates. Sunlight provides the energy through the light-capturing stages of photosynthesis. Carbon collection and light-powered sugar production happen at different times. The leaf gathers carbon in darkness and uses it after sunrise. This daily pattern can be measured. Pineapple leaves become more acidic through the night as malic acid accumulates. Their acidity falls during the day as the stored acid is broken down and its carbon is released. Researchers can follow the rise and fall by sampling leaf tissue at different hours. The pattern forms a chemical record of the plant's nighttime activity. Timing is controlled partly by the pineapple's circadian clock. A circadian clock is an internal system that coordinates activity across a cycle of about 24 hours. Genes involved in stomatal movement and acid storage become active according to this internal schedule. Other genes become more active when daylight arrives and the stored carbon is released. Studies of the pineapple genome show that CAM developed largely through changes in when existing plant genes were used. Evolution adjusted the timing and strength of their activity. That altered schedule allows pineapple to produce more growth from a limited supply of water. Across a field after sunset, the plants may appear completely still. Microscopic pores are opening inside the waxy leaves as carbon begins moving into storage. By morning, the stored reserve is ready to support another day of sugar production. Those small daily gains accumulate over many months. The plant adds new leaves, enlarges its hidden stem, and builds the reserves needed before its first flower can appear. The sugars produced during daylight move out of the leaves through a network of living tubes called phloem. Phloem carries dissolved sugars from the parts of a plant that produce them toward tissues that are growing or storing energy. In a young pineapple, much of that supply is directed into roots, new leaves, and the thick stem hidden inside the rosette. This stage is called vegetative growth. During vegetative growth, the plant builds its body without producing flowers. A new leaf begins as a tightly folded blade near the center of the rosette. It gradually lengthens, hardens, and bends outward as younger leaves appear behind it. Each healthy leaf adds more surface area for collecting sunlight and more cells capable of producing sugar. A larger rosette can therefore support a larger reproductive structure when flowering finally begins. Much of the plant's foundation remains below ground. Pineapple roots are generally shallow, with many growing through the upper layers of soil where rainwater and nutrients first arrive. A mature plant may spread fine roots outward beyond the narrow base of its stem, although most remain far closer to the surface than the deep roots of a large tree. These roots need both moisture and oxygen. When soil remains filled with water, air spaces disappear, and the roots struggle to carry out normal respiration. Wet conditions also favor organisms that can damage the roots and the plant's central tissues. Commercial pineapple fields are therefore commonly established in loose, well-drained soils. Raised beds and drainage channels may be used in places that receive heavy tropical rain. Pineapple also grows well in mildly acidic ground. A soil pH between roughly 4.5 and 6.5 is commonly suitable. The pH scale measures how acidic or alkaline a substance is, with lower numbers representing stronger acidity. Within that range, the roots can obtain important nutrients, while some soil diseases remain less active than they would under different conditions. Water and dissolved minerals enter through the roots and move upward into the leaves. Nitrogen becomes part of proteins and chlorophyll, the green pigment that captures light. Magnesium sits near the center of each chlorophyll molecule. Potassium helps regulate water balance and supports the movement of sugars through the plant. These materials gradually become part of a working system, assembled over many months. A pineapple does not reach flowering readiness on one fixed birthday. Its progress depends on the cultivar, temperature, sunlight, nutrition, water supply, and the type of shoot used for planting. A crown taken from the top of a fruit often begins smaller than a well-developed sucker growing near the base of an older plant. Slips, which form close to the fruiting stalk, may begin at another size and grow at a different rate. Under warm commercial conditions, growers may allow roughly 10 to 14 months of vegetative growth before attempting to induce flowering. Smaller planting material, cool weather, drought, or poor nutrition can extend the wait beyond a year. Growers often judge readiness by examining the plant itself. One useful feature is the D-leaf, usually the longest mature leaf on the plant. Its length, weight, and condition provide clues about the size and strength of the rosette. The number of healthy leaves and the width of the plant also help show whether it has enough capacity to support a marketable fruit. Sugars produced each day are used immediately in some tissues and stored temporarily in others. The thick stem can hold carbohydrates that may later be redirected toward the flower stalk and developing fruit. Minerals remain built into enzymes, cell membranes, pigments, and strong leaf fibers. Long before the first flower opens, the plant has already created the roots that will gather water, the leaves that will supply sugar, and the transport tissues that will connect them. A field of mature plants may appear ready at nearly the same time. Natural flowering can still begin unevenly. One rosette may respond to cool nights or internal hormone levels before the plant beside it. That difference could eventually spread harvests across many weeks and produce fruit of uneven size. Once enough growth has accumulated, a small chemical signal can reach the protected center of the rosette and redirect its growing point toward more than 100 flowers. Protected among the youngest folded leaves is a small dome of living tissue, only a few millimeters across. This is the shoot apical meristem, the cluster of rapidly dividing cells at the pineapple plant's growing tip. For many months, those cells have produced one leaf after another. Each new leaf began near the center, then lengthened and moved outward as still younger leaves formed behind it. Once the plant reaches sufficient size, activity inside this tiny growing point can change within days. Genes that supported ordinary leaf production become less active. Other genes begin directing the formation of a flower stalk, floral bracts, and the individual flowers arranged around it. This change is called floral induction. The new structure remains hidden at first because it begins deep inside the rosette. From outside, the pineapple plant may appear unchanged, while its central growing point has already entered a new stage of life. One of the most important chemical signals involved is ethylene. Ethylene is a plant hormone that exists as a gas. A hormone is a chemical messenger produced in one part of an organism that influences cells elsewhere, often in very small amounts. Plants use ethylene during several processes, including fruit ripening and responses to injury. It also helps control aging in leaves and flowers. Pineapple plants are especially sensitive to ethylene, when they are mature enough to reproduce. Cool periods, shorter daylight hours, drought stress, and changes in the plant's internal chemistry can encourage natural flowering. These influences vary with the cultivar and the condition of the rosette. A large, healthy plant may respond strongly, while a smaller neighbor exposed to the same weather may continue producing leaves. Natural variation creates a problem in commercial fields. If thousands of plants begin flowering on different dates, their fruits will also reach harvest at different times. Some pineapples may already be approaching full size while nearby plants have only begun forming their flower stalks. Harvest crews would need to return repeatedly. Packing facilities would receive uneven amounts of fruit, and shipping plans would become harder to coordinate. Growers learned to reduce this uncertainty by applying flowering treatments to plants that had reached the desired size. One widely used compound is ethaphon. After ethaphon enters plant tissue, it breaks down and releases ethylene. The hormone then travels through small spaces between cells and binds to receptor proteins. These receptors act like chemical switches. Once activated, they begin a chain of signals that changes which genes are used inside the meristem. Older methods have included treatments that generate acetylene, a gas with a molecular shape similar enough to ethylene to affect related signaling pathways. The exact material and application method vary among countries, farms, and production systems. Growers may pour a measured solution into the center of each rosette, or apply it across a field using carefully controlled equipment. The treatment does not create a visible pineapple overnight. Its purpose is to bring physiologically ready plants across the same developmental threshold. Several weeks may pass before the emerging flower structure becomes easy to see. The eventual harvest still lies months ahead. Even so, aligning the beginning of flowering allows fruit age to remain much more uniform across the field. Workers can estimate when plants will need inspection and when harvest volumes are likely to rise. Packing lines can prepare for a more concentrated supply. Refrigerated containers and transport schedules can be arranged before most fruits have reached their final size. Control also involves preventing the signal from arriving too early. A young plant that flowers before developing enough leaf area may produce a smaller fruit. Unexpected cool weather can cause some plants to begin reproductive growth outside the planned schedule. Cultivars differ in their tendency to flower naturally, so growers watch plant size and local weather closely. Some management systems use approved growth regulators to reduce premature flowering during vulnerable periods. The aim is careful timing. A mature rosette must gather enough resources before the chemical message redirects its growing point. Inside that protected center, ethylene-related signals begin reorganizing the plant at a scale too small to see. The dome that once produced long green leaves starts building a thick central stalk crowded with floral tissue. As it rises through the rosette, more than 100 separate flowers begin taking their places around the same narrow axis. Within several weeks of floral induction, the center of the rosette begins to change shape. A thick stalk pushes upward through the tightly folded young leaves. Its tissues are pale and compact at first because they developed away from direct sunlight. As the surrounding leaves separate, the upper part of the stalk becomes visible. At its tip sits the inflorescence, the complete cluster of flowers carried on one reproductive shoot. This dense structure may eventually measure roughly 20 to 30 centimeters, before its tissues enlarge fully into the familiar fruit. Its surface is already divided into many small positions. Each position belongs to one flower. A cultivated pineapple commonly produces between 100 and 200 flowers around the same central axis. Their arrangement is extremely compact, leaving very little open space between neighboring buds. Each flower rests above a fleshy bract. A bract is a modified leaf associated with a flower. The bracts may appear green, yellow, reddish, or pink, depending on the cultivar and stage of development. They protect the young floral tissues and later become part of the hard patterned surface of the pineapple. As the inflorescence develops, individual buds begin extending beyond their bracts. A pineapple flower is small compared with the fruit it helps create. Its petals may be violet, blue, reddish purple, or a shade between them. Many are only a few centimeters long. The flower usually contains 3 sepals, which form the protective outer covering of the bud. Inside them are 3 petals. 6 stamens produce pollen, with each stamen carrying a small pollen-bearing structure called an anther. At the center is the pistil, the female part of the flower. Its upper surface receives pollen. A narrow style extends downward toward an ovary divided into 3 chambers. The ovary sits below the visible petals and sepals. Botanists describe this arrangement as an inferior ovary. The word inferior refers only to its position beneath the other flower parts. That low placement will matter later because the ovary can enlarge while the bract and surrounding tissues remain attached above it. Flowering spreads across the cluster in an ordered sequence. The lowest flowers commonly open first. Over the following days, additional flowers open farther around and higher along the central axis. Several may be visible at once, while younger buds remain closed above them, and older petals begin fading below. The complete flowering period often lasts for several weeks, depending on temperature, cultivar, and growing conditions. Each blossom remains open for only a brief portion of that time. A field at this stage may contain thousands of purple flowers held close to the ground. They are partly hidden among leaves that can reach almost 1 meter in length. The flowers attract far less attention than the heavy golden fruits that will replace them. Yet the entire outer pattern and much of the flesh are being organized during this short stage. The order in which the flowers appear also preserves the geometry established by the growing tip. New floral units were added around the axis at changing angles. Their close placement creates diagonal rows that will remain visible as spirals across the mature rind. For now, the surface is softer. Petals project from between colored bracts, and each flower still exists as a complete reproductive unit. Pollen released from anthers can reach the sticky surface at the center of a flower. Under ordinary circumstances, a pollen grain may produce a tube that grows downward through the style. That tube can carry reproductive cells toward one of the ovules inside the ovary. Successful fertilization would allow an ovule to develop into a seed. With more than 100 flowers, the cluster appears capable of producing a fruit crowded with seeds. Cultivated pineapples usually follow a much quieter reproductive path. Their flowers remain functional, their ovaries continue growing, and the finished fruit commonly contains almost no mature seeds at all. A pollen grain resting on the stigma carries only half of the genetic material needed to create a seed. The stigma is the sticky surface at the center of the flower. When pollen is accepted there, it absorbs moisture and begins growing a narrow tube. That pollen tube extends through the style, the slender passage leading toward the ovary beneath the petals. Inside the ovary are ovules, each containing a female reproductive cell. If the tube reaches an ovule, it releases reproductive cells that join with material from the flower. This process is called fertilization. The fertilized ovule can then develop an embryo, a protective seed coat, and a small supply of stored food. In many flowering plants, this sequence allows one generation to produce the next. Pineapple flowers contain all the basic structures required for it. Successful fertilization still depends on whether the flower accepts the pollen that arrives. Many cultivated pineapple varieties have a genetic system called self-incompatibility. This system allows the flower to recognize pollen carrying certain closely related genetic signals. When the pollen is too similar to the receiving plant, its tube may stop growing before reaching an ovule. The flower remains physically complete and capable of reproduction. Its recognition system simply limits which pollen can finish the journey. Self-incompatibility encourages pollen exchange between genetically different plants. Seeds produced through such crosses receive a new mixture of traits from two parents. That variation can help wild plant populations respond to disease, changing weather, and other pressures over many generations. A commercial pineapple field creates very different conditions. Growers commonly plant thousands of clone shoots from one selected variety. The plants may have come from crowns, slips, suckers, or tissue culture. Yet their inherited information remains nearly identical. Pollen moving from one plant to the next may therefore encounter the same genetic barriers it would face within the flower that produced it. Pollination may also be limited by the animals present around the field. Hummingbirds can pollinate pineapple flowers in parts of tropical America. Certain insects may transfer pollen as well. Commercial production regions outside the plant's original range may have fewer effective visitors, and growers usually prefer to avoid seed formation. In Hawaii, the importation of hummingbirds has historically been prohibited partly because these birds could increase seed development in cultivated pineapples. A mature pineapple seed is small, dark brown, and hard. Even a few can interfere with processing or create an unpleasant surprise in the soft flesh. Most grocery store pineapples therefore contain no obvious mature seeds. Their ovaries still enlarge after flowering. This kind of seedless fruit growth is called parthenocarpy. Hormones within the flower cluster stimulate cell division and expansion, even when fertilization has not produced an embryo. Each ovary begins becoming a fruitlet while its ovules remain undeveloped. The surrounding bracts, sepals, and central stem respond to growth signals as well. Water enters the expanding cells. Sugars arrive through the plant's transport tissues. New cell walls are built as the young fruit increases in size. The plant can therefore invest heavily in fruit tissue, without completing the sexual process that would normally fill that tissue with seeds. Seedlessness makes the pineapple easier to slice, can, and eat. It also allows the flesh to remain more uniform from the rind toward the core. For crop breeders, however, seeds remain essential. Breeders deliberately place pollen from one compatible variety onto flowers of another. The resulting seedlings contain new genetic combinations. Some may inherit greater sweetness, stronger disease resistance, smoother leaves, or fruit that survives shipping more successfully. Most seedlings will not combine every desired quality. promising individuals must be grown, tested, and compared before one is selected for cloning. Commercial farming then returns to vegetative reproduction. A chosen plant can be copied many thousands of times, preserving the traits that made it useful. Sexual reproduction creates variation for future varieties, while cloning supplies the predictable fruit found across present-day fields. As flowering ends, the violet petals dry and withdraw between their bracts. Very few ovules become seeds, yet the ovaries beneath them continue swelling. Each fruitlet presses more closely against its neighbors until the narrow spaces between more than 100 separate flowers begin to disappear. Beneath each fading flower, the ovary begins to swell into a small fruitlet. At first, every fruitlet still belongs to its own floral unit. Its tissues are surrounded by the bract beneath it, the sepals above it, and the neighboring fruitlets packed tightly against its sides. There is very little open space inside the developing cluster. The original flowers were arranged closely around the same central axis, so every fruitlet enlarges within a crowded structure. As their cells divide, neighboring tissues press more firmly together. The outer walls that once separated one flower from the next become thinner and less distinct. This process begins only a short time after flowering. During the earlier stage, growth depends heavily on cell division. One cell produces two. Those cells divide again, and the amount of living tissue increases rapidly. Later, the pace of cell division slows, while individual cells begin expanding. Water enters through thin membranes and fills the large internal spaces called vacuoles. Sugars carried from the leaves accumulate in the growing cells. Organic acids, pigments, enzymes, and structural materials are deposited as the fruit gains weight. The ovaries form only part of this expanding mass. The fleshy bracts beneath the flowers enlarge alongside them. These bracts began as modified leaves, but their tissues remain attached as the fruitlets grow. The three sepals that once protected each flower bud also become part of the developing surface. Additional tissue around the ovaries thickens until the original floral units are joined through broad areas of living material. At the center, the inflorescence axis expands as well. This was the thick stem that carried all the flowers. As the pineapple matures, it becomes the firm, fibrous core extending through the middle of the fruit. Its vascular bundles continue transporting water and dissolved nutrients into the surrounding tissues. These bundles are the pale, thread-like structures sometimes visible when a pineapple is cut lengthwise. The core is firmer because it contains more supporting fibers and vascular tissue than the softer flesh around it. The fruitlets remain connected to this central supply route throughout their development. Their close arrangement causes the entire cluster to enlarge as one coordinated structure. The boundaries between them become compressed and buried, rather than opening into deep gaps. Small seams may remain near the rind, yet the flesh farther inside grows into an almost continuous yellow mass. Botanists call the finished structure a syncarp. A syncarp is a multiple fruit produced when many individual fruits and their associated tissues unite. The pineapple, therefore, contains more than a collection of small berries resting beside one another. Its fruitlets share the same central axis and become joined through their expanding walls, bracts, sepals, and surrounding tissues. By maturity, separating one complete fruitlet from the others would tear through tissue that has become part of the larger body. The connection is especially clear near the core. Each fruitlet, narrows inward toward the central axis, much like a wedge pointing toward the center of a wheel. As more than 100 of these units enlarge together, their inner regions meet around the thickening core. Their outer regions form the rind. Their softer middle tissues contribute much of the flesh that is eaten. The original flowers have now changed so completely that their petals and reproductive parts are difficult to recognize. Their arrangement has not vanished entirely. At the surface, each floral unit leaves behind one raised area, commonly known as an eye. The center of the eye marks the position where one flower once opened. The harder edges around it include tissue from its bract and neighboring units. Grooves remain between the eyes because fusion near the surface is less complete than it is deeper inside. The pineapple, therefore, carries two records of its development at once. Its interior shows how closely the fruitlets joined. Its rind preserves where each separate flower began. Those eyes are arranged in diagonal paths that wrap around the fruit, following the exact geometry established when the flower cluster was still forming. One raised eye on the rind marks the place where a single pineapple flower once opened. At its center is the hardened surface of the fruitlet that developed beneath that flower. The surrounding edges include tissue from the fleshy bract that supported it, and from nearby floral parts that expanded during growth. Shallow grooves separate the eye from its neighbors. Those grooves follow the compressed boundaries where individual fruitlets once met before joining more deeply inside the pineapple. Viewed from a short distance, the eyes may seem scattered across the rind. Following them with the eye reveals a much more ordered arrangement. One diagonal row curves upward around the fruit in a gentle spiral. A second set crosses it from the opposite direction. Together, these lines create the repeating diamond pattern that makes a pineapple recognizable even when only part of its surface is visible. Many pineapples show 8 clear spirals turning in one direction and 13 turning in the other. Larger or differently shaped fruits may reveal additional rows, sometimes including 21 less obvious spirals. The visible count can change with the cultivar, the width of the fruit, and how evenly its fruitlets developed. A long, narrow pineapple may stretch the pattern vertically. A rounder fruit can make the crossing rows appear tighter. The pattern began long before the rind became hard. It was established while the flower cluster was still developing at the center of the leafy rosette. The arrangement of repeated plant structures around a growing stem is called phyllotaxis. Leaves can follow a phyllotaxic pattern, and so can flower buds, scales, or seeds. At the pineapple's growing tip, each new floral unit formed at a slightly different angle from the one before it. The next flower did not sit directly above the previous flower. Its position was shifted around the central axis, leaving room for additional flowers to form without creating straight, crowded columns. In many plants, this shift is close to 137½ degrees. That angle is sometimes called the golden angle. It allows repeated structures to spread around a growing center with relatively even spacing. Small differences in growth can alter the finished pattern, so a living pineapple rarely displays the precision of a printed diagram. The basic rule still places more than 100 flowers around a narrow central stalk without wasting much surface area. Similar developmental geometry appears elsewhere in plants. Sunflower seeds form crossing spirals because new florets are added around the expanding center of the flower head. The scales of a pine cone follow diagonal rows created as the cone's growing tip produced one scale after another. Pineapple eyes preserve the same kind of growth record, although each eye later became part of a large fused fruit. The spiral arrangement also affects how the pineapple expands. Every fruitlet is surrounded by neighbors, positioned slightly above, below, and to either side. As they swell, pressure is distributed around the central axis, rather than concentrated along a few straight seams. Their outer surfaces harden into the rind while their inner tissues grow toward the thick core. The result is a strong shell built from many interlocking units. Near the lower part of the fruit, the eyes are often broader and more developed, because flowering and fruit growth commonly began there first. Farther upward, the pattern continues through units that opened later. The diagonal rows therefore preserve both position and sequence. They show how the flower cluster wrapped around its stem and how development progressed along it over time. Each spiral climbs toward the narrowing top of the pineapple. The eyes become smaller and more tightly packed as they approach the summit. Their rows finally disappear beneath a ring of stiff green leaves. The central axis that carried the flowers continues through the fibrous core and rises beyond the fused fruit, where its living growing point remains visible as the pineapple's crown. Within the crown, the youngest leaves remain folded tightly around a small growing point. This growing point contains meristematic tissue, a region of cells that can keep dividing and producing new plant structures. The crown commonly carries several dozen stiff, pointed leaves. Their compact arrangement resembles a smaller version of the rosette growing at ground level. Each leaf emerges from the same living shoot that continued upward after the flower cluster formed. The pineapple's fruit, fibrous core, and crown, therefore, belong to one continuous structure. During development, the central axis carried the flowers around its middle, while its upper tip continued making leaves. As the fruitlets fused and enlarged below, that leafy tip remained active above them. This is why the crown stays green even as the rind turns yellow and the flesh becomes sweet. Its cells still contain chlorophyll and continue carrying out photosynthesis. The crown also contains dormant root-forming tissues near its base. When placed under suitable conditions, these cells can begin producing new roots. The shoot can then establish itself as an independent pineapple plant without passing through a seed stage. This form of reproduction is called vegetative propagation. It allows a new plant to grow from a living piece of the parent. The crown is only one of several shoots capable of doing this. Small leafy growths called slips may form near the base of the fruit or along the upper part of the stalk. They develop from buds positioned between the fruit and the main rosette. Lower on the plant, larger shoots called suckers can emerge from the short stem or from areas close to the soil. Each sucker contains its own growing point and a set of young leaves. After removal and replanting, it can produce roots, enlarge into a rosette, and eventually form another flower cluster. Growers choose among crowns, slips, and suckers according to the kind of crop they want to establish. A crown taken from a harvested fruit may be relatively small. It must first produce roots and build a wider rosette before it reaches flowering size. A mature sucker often begins with more stored energy and a larger group of leaves. Under similar conditions, it may establish faster and reach reproductive maturity sooner. Slips commonly fall somewhere between those two starting points. although their growth depends on size, variety, and field conditions. Commercial farms sort planting material before it enters the soil. Shoots of similar weight and length may be grouped together. This helps neighboring plants grow at a more even rate. Uniform planting material makes later flowering treatments more predictable and reduces large differences in fruit size. A field planted with a mixture of tiny crowns and large suckers would contain plants at very different stages, even if they all entered the soil on the same day. Vegetative propagation also preserves the selected plant's genetic traits. A crown or sucker usually carries nearly the same inherited information as the parent plant. Its future fruit is therefore more likely to resemble the pineapple from which that line was chosen. Seedlings create more variation because they combine genetic material from two parents. Clone shoots provide the consistency needed for large fields producing fruit with similar shape, sweetness, and harvest timing. A healthy crown from a grocery store pineapple can sometimes produce roots when kept warm and given enough moisture. Success depends on the condition of the crown, the climate, and how long the fruit spent in storage. Rooting may take several weeks, while the wait for a new fruit can last 2 years or longer in a cool indoor setting. The important detail lies in what that growth reveals. The leafy top remains a functioning shoot after the fruit has been harvested. Its cells can rebuild the roots and rosette needed to begin the entire cycle again. Beneath that living crown, the developing fruit follows a different path. Sugars arriving from the parent leaves begin collecting inside its cells. Organic acids shift in concentration. Green pigments slowly break down, and new aroma compounds appear as the fused flower cluster approaches maturity. Running through the firm core are narrow bundles of transport tissue connected to the leaves below. One part of this network is called phloem. Phloem carries dissolved sugars from places where they are produced toward places where they are needed. During daylight, the leaves use carbon dioxide, water, and light energy to build carbohydrates through photosynthesis. Some of those carbohydrates support the leaves themselves. A growing share travels toward the developing pineapple. Plant scientists describe an organ that draws in and uses these materials as a metabolic sink. Young leaves can act as sinks while they are still expanding. Growing roots also require imported sugar. Once the flower cluster begins swelling, the pineapple becomes one of the strongest sinks on the entire plant. Sugars enter through vascular bundles that branch outward from the central core. They move into tissues formed from the fruitlets, bracts, and surrounding floral parts. Inside the cells, some sugar is used immediately to provide energy for growth. The rest can remain dissolved in cellular fluid or be converted into other compounds. 3 sugars make up much of the pineapple's sweetness. Sucrose is the same main sugar found in table sugar. Glucose and fructose are simpler sugars that the plant can use directly during metabolism. Their amounts change as the fruit develops. Cultivar, temperature, sunlight, water supply, and harvest maturity all influence the final mixture. Chemistry can also vary from the lower part of one pineapple to the upper part. The flowers near the base commonly began developing earlier, so those tissues may reach a different stage before the later forming fruitlets near the crown. Sweetness alone does not create the full flavor. Pineapple flesh contains organic acids that produce its sharp, bright taste. Citric acid usually makes the largest contribution. This is the same acid strongly associated with lemons and limes. Malic acid is also present and has a softer sourness, familiar from apples. Smaller amounts of other acids and flavor compounds add to the final result. 2 pineapples can contain similar amounts of sugar and still taste quite different. The fruit with greater acidity may seem sharper and less sweet, even when its sugar concentration is nearly the same. Growers and packing facilities often estimate dissolved material in the juice using a measurement called degrees Brix. A small instrument called a refractometer can measure how strongly a drop of juice bends light. More dissolved material changes the path of that light and produces a higher reading. Because sugar forms a large portion of the dissolved solids in pineapple juice, degrees Brix provides a useful estimate of sweetness. It does not count sugar molecules directly. Acids, minerals, and other dissolved substances also affect the reading. Depending on cultivar and maturity, pineapple fruit may measure roughly 11 to 18 degrees Brix. A reading near the lower end can belong to fruit harvested early or grown under less favorable conditions. Values near the upper end are often associated with sweeter fruit, although acidity still shapes how that sweetness is experienced. Acidity is commonly measured through a method called titration. A laboratory worker adds a liquid of known strength to a juice sample until the acids have been neutralized. The amount required provides an estimate called titratable acidity. Pineapple values may fall roughly between 0.5 and 1.6%, depending on variety and stage of maturity. A fruit with 14 degrees Brix and low acidity may taste mild and sweet. Another, with the same Brix reading, can taste much sharper when its acidity is higher. For this reason, growers often consider the relationship between soluble solids and acidity rather than relying on either number alone. As harvest approaches, sugar continues arriving from the leaves while acid levels and cell chemistry shift. The flesh becomes juicier as expanding vacuoles fill with water and dissolved compounds. Cell walls gradually change, reducing some of the firmness found in young green fruit. Months of nighttime carbon capture and daytime photosynthesis are now stored in a form that can be tasted. Before the pineapple is cut and its juice is measured, those internal changes begin showing themselves outside the flesh. Green pigments weaken across the rind. Golden colors become easier to see, and volatile molecules begin escaping into the air as the fruit develops its familiar scent. Across the rind, the first visible changes often begin near the base of the fruit. The green surface gradually takes on patches of yellow or gold that may spread upward toward the crown. This color shift begins as chlorophyll breaks down. Chlorophyll is the green pigment that allows plant tissues to absorb light for photosynthesis. Young pineapple fruit contains enough of it to keep much of the rind green. As the fruit matures, enzymes dismantle chlorophyll molecules and reduce the strength of that green color. Pigments called carotenoids then become easier to see. Carotenoids produce many yellow and orange colors found in plants, including those in carrots, pumpkins, and ripe pineapple flesh. Some were present while the fruit was still green, hidden beneath the stronger chlorophyll. Others increase as the fruit develops. The change does not always spread evenly. Fruitlets near the base commonly began developing before those closer to the crown, so the lower part may color first. One side can also receive more direct sunlight than the other. Differences in temperature, leaf shade, and the fruit's position within the field may create a rind with both golden and green areas at the same time. Cultivar also matters. Some pineapple varieties develop a rich yellow shell at maturity. Others can retain considerable green color while containing flesh that is already sweet enough to harvest. The fruit also begins releasing a more noticeable scent. Aroma reaches the nose through volatile compounds. The word volatile means that a substance can evaporate easily and enter the surrounding air. Once inhaled, these molecules reach receptors high inside the nasal passages. The brain combines signals from many receptors into the familiar smell recognized as pineapple. No single molecule produces the complete aroma. Pineapple scent is created by a mixture containing esters, alcohols, sulfur-containing compounds, and several other chemical groups. Esters often provide sweet or fruity notes. Some sulfur compounds are present in extremely small amounts, yet have a strong effect on the overall scent. Changing even one part of the mixture can make one variety smell noticeably different from another. These aroma molecules arise through active metabolism inside the fruit. Enzymes convert fatty acids and amino acids into smaller compounds. Sugars and other stored materials also supply ingredients used in aroma-producing reactions. As maturity advances, the amount and balance of these volatile substances change. A freshly cut ripe pineapple releases them quickly because slicing opens thousands of cells and exposes juice to the air. Temperature strongly affects the process. Warm conditions speed many chemical reactions and cause volatile molecules to evaporate more quickly. A pineapple kept warm may become fragrant sooner, although it may also lose aroma compounds and moisture faster. Cool storage slows those changes and extends useful storage time. Excessive cold can damage tropical fruit tissues, so commercial storage must remain within a carefully controlled range. Color and scent provide useful clues, yet neither gives a perfect measurement of sweetness. A pineapple can turn more yellow after harvest while gaining little additional sugar. Ethylene treatment may encourage external degreening, which means the loss of green color from the rind. The treatment can improve appearance without recreating the supply of carbohydrates that once arrived from the plant's leaves. Warm storage can also increase yellowing, while the amount of sugar remains nearly unchanged. Growers, therefore, combine several observations before harvest. They may record the number of days since flowering was induced and examine fruit size or shell color. Sample fruits can be cut to inspect flesh development and internal damage. Juice may be tested for soluble solids with a refractometer, then measured for acidity. The required maturity also depends on the destination. Fruit sold near the farm can remain attached longer. A pineapple intended for a distant market must still be firm enough to survive packing, refrigerated transport, and handling before reaching a store. The final decision balances flavor with the time the fruit must remain intact. That timing carries unusual importance because the pineapple's connection to its sugar-producing leaves ends at harvest. Several days on a counter can alter its color, softness, and scent, but they cannot supply the sweetness that was missing when the stalk was cut. When the harvest knife cuts through the stalk, the pineapple's supply line closes. Until that moment, sugars produced in the leaves could travel through phloem tissue and enter the developing fruit. Once detached, the pineapple must depend on the water, acids, sugars, and other compounds already stored inside its cells. Its main sweetness has largely been decided. Pineapple belongs to a group described as non-climacteric fruit. The term refers to the way a fruit behaves after it leaves the plant. Bananas, tomatoes, and avocados are climacteric. After harvest, they experience a strong rise in respiration, which is the controlled release of energy from stored compounds. Their production of the plant hormone ethylene also increases. That chemical surge activates enzymes involved in softening, color change, aroma, and the conversion of stored starch into sugar. A firm green banana can therefore become much sweeter while sitting away from its plant. Pineapple follows a different pattern. Its post-harvest respiration does not rise through the same large climacteric wave. The flesh also contains little stored starch that could later be converted into enough sugar to transform an underripe fruit. Chemical activity continues inside the harvested pineapple, but no process replaces the months of carbohydrates delivered by the parent leaves. The rind may still turn from green toward yellow. Chlorophyll continues breaking down, allowing golden pigments to become more visible. Acidity can decline gradually, which may cause the fruit to taste milder. Cell walls weaken as enzymes alter substances that help hold plant cells together. This softening can make the flesh seem riper, even when the total amount of sugar has changed very little. Aroma changes as volatile compounds are produced, released, or lost into the surrounding air. Water also escapes through the rind and through the cut base. Over time, moisture loss can reduce weight and leave the crown or outer tissues looking dry. These changes affect eating quality, yet they do not create a major new supply of sweetness. An underripe pineapple may become softer and more fragrant, while remaining less sweet than one allowed to mature longer on the plant. This biology also explains a familiar kitchen suggestion involving storage position. Turning a pineapple upside down does not cause meaningful new ripening. Gravity cannot pull enough sugar through intact fruit tissue to convert a sour pineapple into a sweet one. Sugar is dissolved inside cells rather than resting as a free liquid at the bottom of the fruit. Cell membranes, walls, and vascular structures limit how those compounds can move after harvest. The lower portion may already taste sweeter than the upper flesh. Flowers near the base of the inflorescence generally opened earlier, giving their fruitlets more time to develop. Transport tissue can also distribute materials unevenly as the fruit grows. Storing the pineapple in a different position does little to erase differences created while it was still attached to the plant. Growers must therefore judge harvest maturity with care. One common minimum target for acceptable eating quality is around 12% soluble solids, with titratable acidity no higher than roughly 1%. Standards vary with cultivar, market, and production region. A fruit intended for nearby sale may remain on the plant longer and be harvested with softer flesh or stronger aroma. Export pineapples need enough maturity to taste good after arrival, while retaining the firmness required for packing and transport. They may spend days or several weeks moving through refrigerated storage, ships, warehouses, and stores. Temperatures are often kept within a range of roughly 7 to 12 degrees Celsius, depending on maturity and expected storage time. Warmer conditions speed deterioration. Excessive cold can cause chilling injury, leaving internal browning that may remain invisible beneath the rind. The harvest date must balance developed flavor with the physical strength needed to survive that journey. A pineapple picked too early cannot manufacture the missing sweetness later. One harvested at a more advanced stage may offer better flavor, but tolerate fewer days of handling. The cut stalk therefore marks a lasting chemical boundary. The leaves have completed their contribution, and the fruit carries forward only what they supplied before harvest. Fresh pineapple still contains active chemistry of another kind. Inside its juice are enzymes capable of cutting proteins, changing the texture of food and contributing to the tingling sensation that sometimes appears after a few bites. Fresh juice spreads across the cutting board as the knife passes through the pineapple's yellow flesh. Mixed into that juice are enzymes known collectively as bromelain. Bromelain is a group of protein-cutting enzymes found in several parts of the pineapple plant. The highest commercial concentrations are commonly extracted from the stem left behind after the fruit is harvested. Related enzymes also occur in the edible flesh and juice. Scientists often distinguish between stem bromelain and fruit bromelain because their mixtures contain somewhat different enzymes. Both belong to a larger class called proteases. A protease is an enzyme that breaks proteins into smaller pieces. Proteins are long chains made from smaller units called amino acids. The amino acids are connected by chemical links known as peptide bonds. A protein's exact chain and folded shape determine what it can do. Some proteins form muscle fibers. Others create connective tissue, carry signals, or build flexible structures such as gelatin. Bromelain speeds a reaction called hydrolysis. During hydrolysis, water is used to split peptide bonds. One large protein chain becomes several shorter fragments. As enough bonds are broken, the protein loses the structure that gave it strength or allowed it to form a stable network. This reaction explains why pineapple has long been associated with tenderizing meat. A marinade containing fresh pineapple juice brings active bromelain into contact with proteins near the meat surface. The enzymes begin cutting proteins in muscle fibers and connective tissue. Short exposure can soften the outer layer and make it easier to chew. The enzymes move only a limited distance into a thick piece of meat, so the strongest effect usually remains close to the surface. Long exposure can go too far. As more protein structures are broken, the outside may become unusually soft or mushy. Commercial meat tenderizers sometimes use purified bromelain in carefully measured amounts for this reason. The same chemistry appears in a bowl of gelatin. Gelatin is made from collagen, a strong protein found in animal connective tissue. Heat changes collagen into shorter protein chains that dissolve in hot water. As the mixture cools, those chains partly reconnect and form a loose, three-dimensional network. Water becomes trapped within that network, creating the soft, solid texture of gelatin. Fresh pineapple can prevent that structure from forming. Active bromelain cuts the gelatin chains before they can build a stable network. The mixture may remain watery even after hours in a refrigerator. Canned pineapple usually behaves differently. During canning, the fruit is heated to temperatures high enough to destroy spoilage organisms and extend storage life. That heat also changes the folded shape of bromelain. An enzyme depends on its precise three-dimensional form to hold and react with its target. When heat unfolds that form, the enzyme becomes denatured and loses most of its activity. Cooked or canned pineapple can therefore be mixed into gelatin, with far less risk of preventing it from setting. Bromelain also contributes to the sensation sometimes felt after eating several pieces of fresh pineapple. The fruit's citric acid can irritate sensitive tissue inside the mouth. Active proteases may briefly affect proteins on the surface of the tongue, lips, and cheeks. Pineapple tissues can also contain microscopic needle-shaped crystals called raphides. These crystals are made largely from calcium oxalate and may add a mild scratching sensation as the fruit is chewed. The strength of the feeling varies with ripeness, variety, serving size, and individual sensitivity. For many people, the tingling fades soon after eating because saliva dilutes the juice and the mouth continually replaces its surface cells. A true pineapple allergy involves an immune response and is different from ordinary temporary irritation. Symptoms such as widespread hives, strong swelling, or difficulty breathing require medical attention. Most familiar tingling comes from the combined effects of acidity, active enzymes, and the fruit's fine physical texture. Heating can quiet much of that chemical activity. The living plant itself continues through a different kind of preservation, using crowns and side shoots to carry almost the same inherited traits into a new generation. Across a commercial field, rows of pineapple plants may look almost identical because many began as living pieces of the same selected plant. This method is called clonal propagation. A clone carries nearly the same nuclear genetic information as its parent. The nucleus is the part of a plant cell that stores most of its inherited instructions. When a crown, slip, or sucker produces roots, it becomes an independent plant without pollination or seed formation. Its genetic traits remain far more predictable than those of a seedling created from two parents. A grower who finds a plant with sweet fruit, smooth leaves, and reliable growth can preserve that combination by multiplying its shoots. The crown is the leafy structure attached to the top of a harvested pineapple. It commonly begins as the smallest of the main planting materials. After planting, it must form roots and enlarge its rosette before it can support a flower cluster. Slips develop near the base of the fruit or along the upper part of the fruiting stalk. They often begin larger than crowns and may reach flowering size sooner. Suckers emerge lower on the short central stem or near the plant's base. A strong sucker may already possess several developed leaves and a supply of stored carbohydrates. Its larger starting size can shorten the time needed to produce another fruit. Growers sort these shoots by type, weight, and length before planting. A field established with similar material tends to grow more evenly. Plants that reach maturity together respond more consistently to flowering treatments. Their fruits also approach harvest at similar times, making it easier to organize field crews, packing lines, and refrigerated transport. The parent plant can sometimes continue producing after its first pineapple is removed. A sucker left attached near the base may use the existing root system and lower stem. It grows into a replacement rosette and later forms another fruit. This secondary production cycle is called a ratoon crop. A ratoon crop often matures sooner than a newly planted crown because the plant does not need to rebuild its entire underground system. The second fruit may differ in size or quality, and disease can accumulate in an older planting. Farmers therefore decide whether the faster second crop is worth keeping or whether the field should be cleared and replanted. Large modern operations can also multiply pineapple plants through tissue culture. A laboratory begins with a very small piece of tissue from a carefully selected plant. The sample is cleaned and placed inside a sterile container on a gel containing sugar, minerals, and plant hormones. Sterile conditions reduce contact with fungi and bacteria that could overwhelm the young tissue. The hormones guide groups of cells into producing many tiny shoots. Those shoots can be separated, encouraged to form roots, and gradually moved from laboratory containers into nurseries. A single selected plant can eventually supply thousands of young plants through repeated multiplication. When properly managed, tissue culture can provide uniform planting material and reduce the movement of certain pests or diseases that might be carried on ordinary field-grown shoots. Careful testing still matters because contamination can occur, and cells grown in culture sometimes develop unexpected changes. Cloning gives pineapple farming its consistency. Similar plants can produce fruit with familiar shape, acidity, color, and storage life. The same consistency creates a biological weakness. A pathogen able to infect one clone may find the same vulnerability repeated across thousands of nearby plants. Extreme heat, flooding, or drought may also affect a genetically uniform field in a similar way. New combinations usually enter the crop through deliberate breeding, where compatible parents produce seeds with reshuffled traits. Plant collections also preserve older cultivars and wild relatives that may carry useful resistance. Modern plantations use laboratories and organized nurseries to repeat selected pineapple plants on an enormous scale. The basic method is much older. Long before commercial varieties had names or sterile containers existed, indigenous growers were saving crowns and side shoots from the plants they valued, gradually shaping the cultivated pineapple through generations of careful selection. The crowns and side shoots carried through modern nurseries belong to a practice that began long before laboratories, shipping containers, or written farm records. Pineapple is native to tropical South America. Botanists have often placed its early domestication somewhere around southern Brazil, Paraguay, and nearby river systems. Possible routes of movement follow great waterways such as the Paraná and Paraguay rivers, which connected forests, wetlands, and human communities across large areas. The exact starting point remains uncertain. Pineapple plants were being selected and transported for generations before anyone left a written description of the process. A crown or sucker can survive a journey and produce a new plant far from the parent population. Over time, cultivated pineapples may become separated by hundreds or even thousands of kilometers from the wild relatives that first supplied their traits. This makes their history difficult to reconstruct. Crops such as wheat and maize often leave large quantities of seed, pollen, or durable remains that archaeologists can compare across time. Pineapple is commonly reproduced from soft living shoots. Its juicy fruit, leaves, and stems decay easily in warm, wet soil. A family could carry one useful crown to a new settlement, grow it there, and continue the same genetic line without leaving many traces that would survive for centuries. The cultivated pineapple is sometimes described as a cultigen. A cultigen is a plant shaped and maintained mainly through human care. Its familiar form reflects long selection, rather than the features of one unchanged wild ancestor. Part of that history remains stored in the differences among living pineapple varieties. Some produce small, strongly flavored fruits. Others have pale flesh or deep golden color. Leaf margins may be heavily armed with spines or nearly smooth. These differences preserve pieces of the variation that growers encountered and selected over many generations. The process did not require formal knowledge of genetics. A grower could notice that one plant produced a larger fruit with softer flesh. Its crown, or strongest sucker, could be saved and replanted. A plant with a very fibrous fruit might be used differently or left out of future planting. Repeated choices gradually increased the number of plants carrying preferred qualities. Larger fruits supplied more food from each plant. Sweeter flesh made the fruit more appealing when eaten fresh or used in drinks. Lower fiber created a softer texture. Reduced seed production made the flesh easier to chew. Leaves with fewer or smaller spines were safer to handle around homes and cultivated plots. Strong crowns and side shoots made successful propagation more likely. Plants that grew reliably under local rainfall and soil conditions were especially valuable. Because each selected shoot preserved most of the parent plant's inherited traits, useful combinations could remain stable through many planting cycles. Indigenous societies were cultivating and exchanging pineapples throughout tropical America before European arrival. Tupi-Guarani, peoples and neighboring communities are closely connected with its early history. The name ananas comes from an indigenous South American word associated with the fruit and later spread into many European languages. Pineapples were eaten fresh when fully developed. Their juice and flesh could also be used in fermented drinks. Long leaves supplied tough fibers that could be separated, dried, and twisted into useful material. Different communities used the plant in ways shaped by local traditions, available crops, and climate. Some uses were practical, while others carried medicinal or social meaning that varied from one culture to another. The modern pineapple, therefore, carries the results of countless small decisions made by growers whose names were never written down. Each saved crown continued one choice. Every replanted sucker preserved a preferred set of traits. Movement between settlements widened the plant's range and exposed it to new soils, rainfall patterns, and human preferences. By the time European sailors encountered pineapples in the Caribbean near the end of the 15th century, the plant had already traveled far beyond its probable South American homeland. Its golden fruit had arrived there through an older network of cultivation and exchange, ready to begin a much faster journey across the oceans. Living pineapple shoots moved northward through tropical America, long before European ships entered the Caribbean. Cultivated plants were already growing across parts of South America, Central America, and several Caribbean islands. This wide range came largely from human movement. A pineapple rarely spreads over great distances through seed alone, especially when cultivated forms produce very few mature seeds. A crown, slip, or sucker could be carried between settlements, planted in warm soil, and used to reproduce the same valued plant. One healthy shoot was enough to establish a new line of pineapples far from the field where it began. River travel helped connect communities across tropical South America. Coastal routes and island voyages extended those exchanges through the Caribbean. People carried crops for food, fiber, trade, and future planting. Pineapple traveled alongside other useful plants within agricultural systems that had already developed over many generations. European records began entering this much older story during the second voyage of Christopher Columbus. In November of 1493, the expedition reached the island of Guadeloupe in the Lesser Antilles. Members of the voyage encountered pineapples there among foods and cultivated plants already known to the indigenous inhabitants. The fruit was unfamiliar to the arriving Europeans. Its cultivation, movement, and selection had been underway for centuries before that meeting. Descriptions of its sweetness and unusual appearance soon traveled back across the Atlantic. Two names followed different routes through language. Ananas developed from indigenous South American words associated with the fruit, especially forms recorded from Tupi-Guarani languages. Portuguese, French, German, and many other languages eventually adopted versions of this older name. The scientific genus also became Ananas, preserving the connection in modern botany. English followed another path. The word pineapple had once been used for the reproductive cone of a pine tree. The tropical fruit's rough surface and repeating scales reminded English speakers of that familiar cone. Over time, pinecone became the usual name for the tree structure, while pineapple remained attached to the fruit. Naming the fruit was easier than transporting it. A ripe pineapple contains a great deal of water and soft tissue. Warm temperatures speed respiration, moisture loss, and microbial growth after harvest. During an ocean voyage that could last many weeks, the fruit might bruise, ferment, or rot before reaching Europe. A fresh pineapple arriving intact became an unusual event. Living planting material offered greater long-term value. Crowns could remain firm after the fruit beneath them had begun deteriorating. Slips and suckers carried stored energy within their leaves and stems. Survival still depended on careful handling. Salt spray could damage exposed tissues. Strong sun and dry wind removed water from the leaves. Too much moisture encouraged rot inside the crowded conditions of a wooden ship. A shoot that survived the voyage needed warm soil and months of growth before it could produce another fruit. Portuguese and Spanish maritime networks carried pineapple planting material into new tropical regions during the 16th century. The plant became established along parts of the western and eastern coasts of Africa. Ships also carried it around the Cape of Good Hope and into ports connected with India and Southeast Asia. Local growers then moved plants inland or between neighboring islands. In regions with warm temperatures, sufficient rainfall, and well-drained soil, pineapple could grow outdoors much as it had in tropical America. Its use changed as it entered new farming systems. Some fruits were eaten near the place of harvest. Others supplied ports, households, and regional markets. Crowns from harvested fruit could be planted again, allowing one imported shoot to become dozens of plants over several growing cycles. This pattern helped pineapple spread without depending on compatible pollen, seed production, or the presence of its original pollinators. By the end of the 16th century, the plant had crossed both the Atlantic and Indian Oceans. Its expansion rested on the same biological trait that indigenous growers had used for generations. A detached shoot could preserve the selected plant and begin again in another place. Warm colonies could place those shoots directly into open fields. Farther north, where frost and cold winters stopped tropical growth, producing a single pineapple required glass, constant heat, skilled labor, and years of careful attention. A living pineapple brought into northern Europe entered a climate far outside the conditions that shaped it. The plant grows best with steady warmth. Prolonged cold slows its metabolism, reduces root activity, and can stop new leaves from forming. Temperatures near freezing damage the soft tissues, while frost may kill the growing point hidden at the center of the rosette. European gardeners therefore had to build an artificial tropical environment around every plant. During the 17th and 18th centuries, wealthy estates began using glass houses and specialized structures known as pineries. Some designs place pineapple plants in deep beds or pits surrounded by heat-producing material. Fresh horse manure releases warmth as microorganisms break it down. Tanner's bark, left over from leather production, could also ferment slowly and provide heat for many weeks. Gardeners packed these materials beneath or around containers holding the plants. More elaborate buildings used furnaces connected to flues running through walls or beneath raised beds. Hot gases passed through the flues and warmed the structure before leaving through a chimney. Glass admitted sunlight during short winter days, while shutters or insulating mats could reduce heat loss after dark. Maintaining warmth solved only part of the problem. A sealed glasshouse could overheat on a sunny afternoon, even while cold air remained outside. Gardeners opened vents and doors to control temperature and humidity. Too little airflow encouraged mold and fungal disease. Heavy watering caused further trouble because cool, saturated soil deprived roots of oxygen and increased the risk of rot. Every plant required careful judgment. Water might be warmed before use, so the roots were not shocked by a cold soaking. The beds had to remain warm without becoming hot enough to damage them. Leaves were inspected for insects, discoloration, and decay. This work continued through many months of vegetative growth before the plant even began forming flowers. One early European success belonged to Agneta Bloch, a Dutch art collector and skilled horticulturist. At her estate near the River Vecht, she grew rare plants arriving through Dutch trade networks. In 1687, Bloch successfully produced a pineapple from a planted slip. The achievement showed that a tropical American crop could complete its full life cycle in the Netherlands when warmth, drainage, and light were carefully managed. Producing a pineapple under these conditions required much more than purchasing one plant. A wealthy household needed land for the garden and glass structures. Fuel had to be supplied through cold seasons. Skilled gardeners watched the plants daily, and replacement shoots depended on continued contact with overseas trade. The finished fruit represented the entire system that had kept it alive. A homegrown pineapple could therefore carry more value as a display than as food. It appeared at the center of banquet tables, where guests could see its patterned rind and tall crown. Some fruits were shown on several occasions before they were finally eaten. Paintings included pineapples beside expensive fabrics and imported objects. Their shape appeared on ceramics, silver containers, carved furniture, and decorative gateposts. By the 18th century, the pineapple had become strongly associated with hospitality. Offering such a rare fruit to visitors displayed generosity because the host was presenting something that demanded unusual expense and labor. The symbol also depended on colonial systems that moved plants, knowledge, wealth, and workers across oceans. Pineapples reached European estates through the same trade routes that carried other tropical crops. Plantation labor and unequal control of land helped supply the expanding market. The image of welcome inside a wealthy home was therefore connected to much larger systems beyond its doors. Architecture made the symbol especially visible. At Dunmore Park in Scotland, an 18th century garden building was crowned with an enormous stone pineapple. The carved fruit rises above the roof with leaves spreading around its base. It transformed the success of tropical cultivation into a permanent monument visible from across the estate. A real pineapple might soften within days, while its stone version could continue announcing wealth for centuries. The fruit's rarity began to fade only when producers could grow enormous tropical harvests and preserve them before decay began. That change would move pineapple away from heated estates and into factories built close to the fields. A tropical field could produce thousands of pineapples, yet distance still stood between those fruits and most consumers. Fresh pineapple is heavy, bulky, and easily bruised. Its cells remain biologically active after harvest, using oxygen and releasing energy through respiration. A hard impact can rupture those cells beneath the rind, creating damaged areas that may darken before anyone notices them. Once the fruit is peeled or sliced, deterioration speeds up. Juice escapes from broken tissue. Oxygen reacts with exposed compounds, and microorganisms gain access to the moist flesh. Warm conditions allow many yeasts, molds, and bacteria to multiply quickly. Before dependable refrigeration, an ocean voyage could last far longer than a ripe pineapple remained in good condition. Canning offered a way to separate the fruit from the short life of its fresh tissues. Workers first removed the crown and thick rind. The pineapple was cored, trimmed, and cut into rings, chunks, or crushed pieces. Prepared fruit entered metal cans with juice or syrup. The containers were sealed and heated to temperatures high enough to destroy many organisms that cause spoilage or disease. Heat also stopped much of the fruit's remaining enzyme activity. Bromelain lost its working shape, which is why canned pineapple no longer cuts proteins as strongly as fresh fruit. The airtight container kept new microorganisms from entering after heating. A pineapple that might soften within days could now remain usable for months or even years. This process made it practical to grow fruit in one tropical region, preserve it close to harvest, and ship it across great distances. Hawaii became one of the most important centers of that new industry. Small-scale pineapple cultivation was already present there during the 19th century. Commercial expansion accelerated near the end of that century as growers acquired land and developed better connections with ports. In 1901, James Dole founded the Hawaiian Pineapple Company on the island of Oahu. Hawaii offered warm weather, productive soils in several regions, and shipping access to the western United States. American investment supplied machinery, canneries, transportation, and large areas of plantation land. One variety became especially useful for processing. Smooth cayenne usually produced a fairly cylindrical fruit with pale yellow flesh. Its eyes were relatively shallow, so less flesh had to be removed during peeling. The shape allowed machines to cut more regular rings, while its size and yield suited large canneries. Uniform fruit could move through equipment designed around consistent measurements. Mechanical peelers removed the rind far faster than hand labor alone. Corers cut out the firm central axis. Blades divided the remaining cylinder into rings of even thickness. Smaller pieces and juice could be directed into other products, reducing some waste from the main process. fields and factories became parts of one connected system. Narrow-gauge railways and field roads carried harvested pineapples toward canneries. Factories required a steady supply because ripe fruit could not wait long in tropical heat. Cans, labels, sugar, fuel, and packing materials had to arrive on schedule. Finished products then moved toward Honolulu and other ports for shipment across the Pacific. This industrial growth depended on a large workforce. Plantations and canneries employed people from Hawaii and migrants arriving from Japan, the Philippines, China, Portugal, Korea, and other regions. Workers planted shoots, removed weeds, harvested fruit, operated machinery, and packed cans. Their labor supported an industry that brought pineapple into ordinary households far from the tropics. The canned ring became common enough to appear in desserts, salads, baked dishes, and military rations. Over time, the same global system that strengthened Hawaii's pineapple industry also weakened it. Companies found regions where land and labor cost less. Production expanded in places including the Philippines, Thailand, and Central America. Long-distance shipping became more efficient, and processing plants could be built closer to newer plantations. By the late 20th century, Hawaii had lost much of its former dominance. Canning had transformed pineapple from a rare display into an affordable stored food. That success favored varieties suited to machines and metal containers. A growing market for whole fresh pineapples demanded a different combination of sweetness, color, firmness, and the ability to remain attractive through weeks of refrigerated travel. A pineapple destined for a can can be processed within hours of harvest. A fresh pineapple faces a much longer journey. It may leave a field in Central America, enter a refrigerated container, cross the Atlantic, pass through a warehouse, and remain on a store shelf before anyone cuts it open. The fruit must taste good after that journey while still looking firm and undamaged. Commercial success depends on several traits working together. Sweet flesh attracts consumers, while moderate acidity keeps the flavor pleasant. A regular shape makes fruit easier to pack. A strong rind protects the softer tissue beneath it. Uniform size allows boxes, pallets, and shipping containers to hold predictable amounts. Resistance to disease matters in the field, and slow deterioration matters after harvest. A pineapple can taste excellent when eaten beside the farm and still perform poorly as an export crop. Soft flesh may bruise during packing. Deep eyes can trap dirt and require more trimming. A crown that breaks easily reduces the fruit's appearance. Uneven ripening can cause some pineapples to decay, while others remain too green. During the 20th century, breeders in Hawaii worked to combine flavor with the demands of international transport. One result was a cultivar known as MD2. A cultivar is a cultivated plant variety, selected for a particular group of inherited traits. MD2 came from the Pineapple Research Institute, an industry-supported breeding program established in Hawaii during the 1920s. Breeders crossed genetically different pineapple plants and raised the resulting seedlings. Each seedling carried a new mixture of genes from its two parents. Some produced sour fruit. Others had weak plants or poor shape. A small number combined useful qualities well enough to receive years of further testing. Promising plants were evaluated across repeated crops and different growing conditions. Researchers measured fruit size, sugar concentration, acidity, color, disease response, and storage life. Once a useful plant had been identified, crowns and side shoots allowed it to be copied rather than recreated from seed. MD2 became widely important during the 1990s. Its flesh was usually golden yellow, with relatively high sugar and lower acidity than many older commercial pineapples. The fruit often developed a regular cylindrical shape and an attractive golden rind. It also remained in marketable condition longer than many varieties that had been bred mainly for canning. These qualities helped MD2 become the dominant pineapple in much of the international fresh fruit trade. Its success also depended on refrigeration. After harvest, the fruit continues respiring, which means its cells keep using stored sugars and oxygen. Cool temperatures slow that activity. Refrigeration also reduces water loss, delays tissue softening, and slows the growth of many microorganisms. Temperature must still remain high enough to protect a tropical fruit from chilling injury. Long exposure to excessive cold can cause internal browning, poor aroma, and damaged flesh that may remain hidden beneath a normal-looking rind. Exporters often keep pineapples within a carefully controlled range near 7 to 12 degrees Celsius, depending on maturity and expected travel time. The cold chain must continue from packing facility to ship, warehouse, and distribution center. A long, warm interruption can shorten the remaining shelf life even after the fruit becomes cool again. Uniform plants make this system easier to manage. Fields grown from similar clones can be treated for flowering at nearly the same stage. Their fruits mature within a narrower period, allowing harvest crews to work through organized blocks. Packing houses receive fruit of similar size and age. Refrigerated containers can then be filled according to shipping schedules planned weeks earlier. By 2024, international exports of fresh pineapples had reached roughly 3.3 million tons. Costa Rica supplied approximately 65% of that trade. The Philippines contributed around 21%. Exact shares change with weather, prices, disease, and production decisions. Yet the overall pattern remains highly concentrated. A large portion of the fresh pineapples crossing international borders comes from only a few regions and from closely related commercial plants. The pineapple on a supermarket shelf is therefore shaped by two forms of selection. Breeders selected its inherited traits, while the supply chain selected for fruit able to survive distance. Its sweetness, color, firmness, and shape all reflect that combined pressure. The resulting uniformity makes global distribution remarkably efficient. It also places the same inherited strengths and weaknesses across enormous fields of nearly identical plants. Across a large MD2 field, thousands of plants may carry nearly identical inherited instructions. That uniformity helps growers predict flowering dates, fruit size, and harvest timing. It also means that a biological weakness present in one plant may be repeated across many hectares. A genetically varied population can contain individuals with different levels of resistance to the same disease. Clonal fields offer a narrower range of responses. Once a pathogen succeeds against one plant's defenses, nearby plants may provide almost the same conditions for infection. The timing of the crop can create a second form of concentration. When most plants flower together, their fruits also approach maturity within a limited period. Heavy rain, a shortage of workers, or a delay at a packing facility can affect a large share of the harvest at once. One pineapple-specific example begins with an insect only a few millimeters long. Mealybugs have soft oval bodies covered with a pale, waxy coating. They gather in protected spaces near leaf bases, around roots, and beneath plant debris. Their mouth parts pierce plant tissue and draw out sugary sap. Several viruses known as pineapple mealybug wilt-associated viruses have been linked with a damaging condition called mealybug wilt. Affected leaves may turn reddish, lose firmness, and begin curling downward. Roots can weaken, reducing the plant's ability to absorb water. Severe cases may leave the rosette unable to support normal fruit development. Ants can make the problem harder to control. Mealybugs release a sugary liquid called honeydew while feeding. Some ants collect this liquid and protect the insects from predators. They may also carry mealybugs into new parts of the plant or between neighboring plants. Management can therefore require clean planting material, control of mealybugs, and attention to the ants that support them. Removing infected plants and cleaning equipment can reduce movement through a field. Other diseases arrive through wet soil. Several species in the genus Phytophthora can attack pineapple roots and the soft tissues near the center of the rosette. These organisms produce swimming spores that move through films of water. Poor drainage gives them better access to roots and lower leaves. Warm, saturated soil can allow infection to spread quickly after heavy rainfall. Root rot reduces the plant's ability to take in water and minerals. Heart rot reaches the central growing point where new leaves and the future flower structure are formed. Damage there can stop the plant from developing a normal rosette or fruit. Raised beds and drainage channels help move excess water away. Growers may also use resistant planting material, approved treatments, and field sanitation, where the disease is common. Vulnerability continues after harvest. A pineapple kept too cold for too long can develop chilling injury. Its rind may still appear acceptable, while the flesh inside begins turning brown or translucent. Temperatures below the safe range disturb cell membranes and alter normal metabolism. The damage may become visible only after the fruit warms or is sliced open. Bruising can remain hidden in a similar way. Pressure during harvesting, packing, or transport may crush cells beneath the rind without breaking the outer shell. Darkened tissue and off flavors develop later. Export standards often reject fruit with severe bruising, irregular shape, crown damage, or internal defects. Some rejected pineapples still contain edible tissue, yet their appearance or storage risk makes them unsuitable for distant markets. At a scale of millions of tons, those losses can represent large amounts of water, fertilizer, fuel, and labor invested in fruit that never reaches a consumer. The production field creates additional material after every harvest. Long fibrous leaves, trimmed crowns, damaged fruit, and thick stems remain behind. Poorly managed waste can shelter pests or add pressure to nearby soil and waterways. Chemical use must also be controlled carefully to protect workers and reduce movement beyond the field. Drainage systems can limit standing water, while carrying soil or dissolved materials away during storms. Ground cover and careful planting help reduce erosion between rows. The modern pineapple system gains efficiency through repetition and concentration. Those same qualities can magnify a disease outbreak, a storage failure, or an unusually wet season. Greater resilience may depend on restoring some of the variety that commercial production removed, while finding useful purposes for the leaves and processing material that each harvested fruit leaves behind. Some of that lost variety survives in older cultivars, wild relatives, and carefully maintained plant collections. Breeders can cross commercial pineapples with these less familiar forms from tropical America. A wild plant may carry resistance to a disease that damages MD2, while an older cultivated type may flower more reliably during unusual weather. Other plants may offer drought tolerance, smoother leaves, firmer flesh, or a flavor worth preserving. These possibilities are protected in germplasm collections. Germplasm is living genetic material kept for future use. A collection may include seeds from compatible crosses, rooted shoots, frozen tissue, and entire plants maintained in fields or greenhouses. Preserving several forms matters because cultivated pineapple is often cloned and may produce few seeds. A rare shoot carrying useful resistance can disappear permanently when the last living plant is lost. Modern genetics helps researchers examine this stored diversity more efficiently. The pineapple genome contains the inherited instructions used to build its leaves, flowers, fruit, and water-saving photosynthetic system. Researchers can compare sections of DNA associated with sugar metabolism, acidity, flowering time, or resistance to disease. They also study genes involved in CAM photosynthesis, the nighttime carbon storage process that allows pineapple leaves to conserve water. Small differences in DNA can serve as genetic markers. A marker gives breeders an early clue that a young plant may have inherited a useful trait. This can reduce the need to wait through many months of leaf growth before every seedling produces a fruit. Promising plants must still be grown under real field conditions. Repeated testing reveals whether their flavor and yield remain useful, while the plants face weather, disease, and storage. Future improvements may also come from using more of each plant. The long leaves left after harvest contain strong fibers built largely from cellulose. Cellulose forms much of the framework in plant cell walls. After softer material is removed, pineapple leaf fibers can be cleaned and processed into thread, paper, reinforcement layers, or composite materials. Some textile systems already turn these fibers into lightweight fabrics. Biocomposites can use them to strengthen panels and molded objects, while replacing a portion of heavier materials. Processing waste contains other possible resources. Stems can supply bromelain for food and industrial uses. Peels and cores contain sugars and organic acids that may support fermentation or carefully prepared animal feed. Other plant material can sometimes become compost or fuel. Practical limits determine whether these uses succeed. Leaves spread across a field can be expensive to collect, and processing equipment must be close enough to reduce transport costs. Residues may contain soil or agricultural chemicals that affect how safely they can be used. Local infrastructure and reliable markets must support the final product. The plant itself still begins with the same quiet processes. Microscopic pores open through the night and collect carbon dioxide while the air is cooler. Months of leaf growth turn that stored carbon into sugars and living tissue. An ethylene signal changes the activity of a growing point only a few millimeters wide. More than 100 flowers then open around one central axis. Their ovaries swell beside fleshy bracts as the stem beneath them thickens into a core. The separate fruitlets gradually become one connected pineapple. human hands have guided that process for generations. Indigenous growers selected useful shoots and carried them through tropical America. Ocean trade moved the plant into new continents, while heated buildings later carried it through European winters. Canning made distant harvest durable, and breeding shaped a pineapple able to cross oceans while remaining firm and sweet. One whole fruit can now rest quietly on a kitchen counter, carrying every scale of that history within its structure. Its crown remains a living shoot. The core preserves the axis that once held a column of flowers. Across the rind, each eye marks one floral unit that began with its own petals, ovary, and place in the spiral. What once looked like a rough, repeating surface has become a clear map. More than 100 flowers opened separately, enlarged toward their neighbors, and slowly joined until their borders became almost impossible to find again.